Okay, so let's go through the synthesis of thyroid hormone. And to give you some context, I'm going to actually detach my screen here so I can draw. Um, it's always good to have like a little window where you're zoomed out so you can know where the tiny complex process is happening. So I'm going to draw that over in the corner. So hopefully you've had a chance to look at the endocrine system histology slides by now. And if you have, what that would mean is you've seen little sort of circles that roughly look like this. So they're small circles made of a specialized simple cuboidal epithelium. And I'm going to just go ahead and put a nucleus in each of these because they have one. And then in the middle is this light pink stuff called colloid. So this picture, the big one, is zoomed in on that. So on the left-hand side, we're seeing blood vessels that go between thyroid follicles. In the middle, we're seeing a follicular cell, and then the lumen is full of colloid. So that's what we're looking at. We've zoomed in on that spot in particular. All right, so I know that this is a very busy image, but as is my practice with stuff like this, where there's a lot of new info and it's intimidating to look at, I'm going to go one step at a time. So the first one I'm going to focus on is actually this right here. So we've got secretory vesicles. So if you remember a little, let's do a tiny bit of cell bio review. A vesicle is just a small bubble of membrane, and it has stuff inside of it, and the bubble is used to transport or excrete whatever's inside of it. So in this case, this step here is making thyroglobulin. So let's break down that word. And I don't have much space to do it, so I'm going to use my margins. So thyro is thyroid. And globulin is a fancy way of saying a protein that is glob-shaped or globular. So it's not that's a way of saying it's not a fibrous protein like muscle fiber or like collagen. It's a globular protein. So it's a rope of amino acids that's been sort of wadded up like a used tissue. So the reason I'm being so specific about this is because students often mistake thyroglobulin for thyroid hormone, and they are not the same thing. So thyroglobulin is there to stabilize and carry the thyroid hormone. And it stabilizes it while it waits in the colloid. So because of that, it's a protein. Globulins are proteins. So here we have an mRNA strand with ribosomes on it. So this is 160 stuff. Remember, mRNA gets translated into proteins. That's all this means. It's just saying, hey, this is a protein, so we have to make it like we make other proteins. So the secretory vesicles here just contain a slightly unfinished thyroglobulin. And as that vesicle migrates to where it's going to pop out, it's a precursor. So the precursor has to be turned in to the final form. And the final form comes with the following steps. So thyroglobulin precursor, this is thyroglobulin. And then we've got our amino acid derived hormone, because if you go back to your hormone classes, thyroid hormone is an amino acid derived hormone, tyrosine specifically. So you got to add iodines to it, which you can see here. And then you need to glue the thyroglobulin onto it as well. So essentially, you've got the protein component being made here. The modified amino acid already exists. So this step here is just gluing all of those things together. And This is hydrogen peroxide. Just like you might have in your bathroom or first aid kit, not different. And the hydrogen peroxide, so like think about what we use hydrogen peroxide for in our regular lives. We use it as a disinfectant. We use it for cleaning in some cases. We use it for teeth whitening. Why do we use it for that? Hydrogen peroxide is good at rearranging covalent bonds, which is what's happening here. So you have to take a precursor protein, glue it to this double amino acid business, and also glue on some iodines. So having hydrogen peroxide there, along with some enzymes, just helps to make sure that that gluing together step happens correctly. So that's all you need to know about that. It's just there to facilitate the construction of the final process. So what you get is exocytosis, shown here, of this vesicle with the now completed thyroglobulin with the thyroid hormone, which here is being called iodinate and couple. Because if you look carefully, you can see two tyrosines with two iodines glued together via an oxygen, creating a complete thyroid hormone. So it just blorps out this finished product. So hormone component with stabilizer component glued on. So this is what you see when you look at the histology image. The pink stuff, this stuff, is just a bunch of thyroglobulin with fixed and not yet usable thyroid hormone inside. And it happens to stain pink because the stain, hematoxylin and eosin, eosin is the pinkish component of that, and it just so happens that thyroglobulin and thyroid hormone like to pick up that pink stain. So this is handy because that means you have a gland that's full of these little bubbles made of cells, and they contain thyroid hormone that you can release at will. So what happens when it's time to release? Well, let's move our attention on down to the lower component. And I want to remind you guys, so this process where thyroglobulin is endocytosed and moves across here, which I will explain, is stimulated by thyroid stimulating hormone. So TSH from the anterior pituitary is what's going to cause this series of events, right? Because if the thyroid is not instructed to do so, it won't really release any of this stuff. So just keep in mind when you see this, that means that the thyroid is being bossed around by the pituitary. So we've got endocytosis by Pseudopodia, which just means pretend foot. That's all Pseudopodia means. That's what we call it when a cell reaches out a part of its membrane and basically noms something up. Let me just really quickly read this question in the chat. So the bubbles are thyroglobulin before it's mixed with other thyroid hormones. Yes, that's exactly right. So these guys up here are thyroglobulin being finished up before it's combined with thyroid hormone to produce this big guy right here. Okay. So one of the important features of thyroglobulin, and there are others, is if you think about what thyroid hormone does, its whole job is to really intensely activate an increase in metabolism. So spend more ATP, burn up oxygen much more quickly. And we need this, but its action needs to be directed. So what you don't want it to do is be acting on the cells that are producing it while it's sitting in storage. So while it's sitting in storage, it's glued to thyroglobulin partially because we want to keep it inactive until we need it. So once it's gobbled up, we have vesicles again, and vesicles are of colloid, so this pink stuff in here. And now what we need to do is break off the thyroglobulin because that's what's preventing the thyroid hormone from doing its job. So lysosomes come in to help with that. And remember, lysosomes are a vesicle inside of a cell that contains proteolytic enzymes and other recycling enzymes. That's kind of the job of the lysosome, is to be the recycling center of the cell. And so basically it goes in and it doesn't care about this because it's not an enzyme that's designed to break apart hormone. It's designed to break apart proteins. So the lysosome combines with the colloid vesicle, and the proteases inside of it go and just basically dismantle the thyroglobulin. So once they're done with that, you get some waste compounds that contain iodine. You don't need to know what these mean. The point here is that these waste products with iodine contribute to a source of iodine that you can use to make new thyroid hormone. So there's a bit of a recycling step here where you can, some of the iodines in this vesicle get removed, and that's how you make T3. So if you look at this, 1, 2, 3, 4, if I break thyroglobulin off of this, it's going to be T4. Some of the T4 in this step down here gets converted to T3 on the way. So an iodine's taken off of it. And where that iodine goes is it gets used to make a new thyroid hormone. So once you take thyroglobulin off and you remove an iodine from some of that thyroid hormone, then you get T3 and T4. Those are exported into the blood. Now, if you go back to your book figure, what you'll see, and I'm going to erase some stuff so I can draw this, is, again, so that thyroid hormone can be carried in the blood, because it's a weird exception. So catecholamines can travel freely in the blood, and they bind to outside receptors, and melatonin is the same. But so I was mentioning other amino acid based hormones. So if you're like, why is she talking about those? It's because I need to mention them in the context of a different transport mechanism, let's say. So most of the amino acid hormones can travel freely in the blood and they are just fine, but thyroid hormone is an exception. So once the T3 and T4 get out, if you look back over at figure 18.11, what you'll see is that there's various carrier proteins waiting to carry the thyroid hormone. And so let me draw thyroid hormone as being a little dumbbell kind of shaped guy. So this is T3/T4. And the carrier protein could be TBG, which stands for thyroid binding globulin, or transthyretin, or even just albumin. So the point of that is, without asking you to memorize the names of all of these carriers, because I don't expect you to memorize all of them, the main idea and the important idea here is that once it's released, T3 and T4 have to be carried by a protein in the blood as well. And then they migrate to the cells and tissues that they target, which is most of your body's cells and tissues. And they are able to enter the cell and go inside and land on the mitochondrion and cause an increase in ATP production. That causes an increase in oxygen consumption, which enables the cell's metabolism and therefore sort of daily task completion to increase in speed. So let's summarize before I close this. Step 1, make a protein and then glue the hormone to it. Once you've done that, you can complete the step using some hydrogen peroxide, and now you have a completed, stable hormone. When it's time to secrete, you take that completed and stabilized hormone and you get rid of the thyroglobulin, creating a free and active T4. Some of that T4 gets converted to T3 because an iodine is removed. And then that T3 and T4 mixture is transported outside into the blood, where it combines with blood protein so it can get carried around. And that is that on thyroid hormone.